As part of the mandate of the Infectious Disease Epidemiology Research Team, we hosted 4 A-level students from the ENAS Hybrid School. We took the students through a 3-day intensive hands-on practical training on molecular biology techniques and bioinformatics. During the training, they had hands-on experience with pipetting, DNA extraction, PCR, sequencing, and bioinformatics.
Hi, my name is Saleh.
Hi, I'm Simone, and we are from ENAS Hybrid.
We had a 3-day trip to KCCR where our main purpose was to learn about gene technology. I learned about PCR and the 2 types of PCR, conventional and real-time, and I learned how to analyze data from a real-time PCR machine.
I had the opportunity to learn more about the micropipettes and how it's used. I learned more on practical skills and how I could apply these skills when it comes to practicing or doing practicals in school.
Our 3-day trip was really beneficial. It helped improve my confidence in my practical skills. I really wish it was longer, but it was quite enough for us to have fun and understand.
Overall, it was a very good experience, and we are looking forward to coming here in the near future.
Hi, I'm Thelma,
Hi, I'm Sinead.
and we are from ENAS Hybrid School.
So we are grateful to have an opportunity to study here at KCCR, where we learned about extraction and how to isolate DNA from a blood sample to test for whether it had a Plasmodium parasite.
Yes, and I also learned about the DNA sequencing, which is a process used to determine the order of nucleotides.